Here to the first of our Midsummer Nights lectures. I'm Eric Lander. I'm the director of the Broad Institute of MIT and Harvard, and I'm delighted to see so many of you coming to join us on a beautiful summer evening. You know, we're living through an extraordinary time in biomedicine right now. It's just a remarkable time, what has been going on. Really, well, tracing back to the discovery of the structure of DNA and the understanding of the genetic code and the development of molecular biology and cloning, much of which took place here in this city. And then in the 1990s, the launch of a human genome project, a coming together in a collaborative fashion of scientific groups around the world to try to get information about all of the human instruction set, all of the genome, and make it freely available on the web so that thousands and thousands of groups around the world could use it to propel progress in science. That was a really major turning point in many ways in the progress of biomedicine. It was a turning point because it produced a huge set of data. It was a turning point also because it produced a change in culture. It was a collaborative project. It could only happen by people coming together across disciplines, across, yes, medicine and biology, but also chemistry and engineering and computer science, and people coming together across institutions and countries. When the Human Genome Project reached its successful conclusion in the early part of this decade, many of us, who I will say were deeply, deeply involved in the Human Genome Project, for those of you, most of you who live here in Boston, Boston was the single largest contributor. Cambridge and Boston were the single largest contributor to the Human Genome Project, with something like a third of the entire Human Genome Project done right within just 2 blocks away at the other location that we have. As that was reaching its conclusion, we all recognized that there was something pretty important, pretty magic about the power of collaboration across institutions, across disciplines, about making information available. And it led to the idea of creating a new kind of institute. The Broad Institute grew from that idea. The Broad is an unusual sort of institute. It cuts across all of Harvard, its medical school and its School of Public Health, and its Faculty of Arts and Sciences, and all of MIT, its engineering, computer science, its biology, chemistry. And it also cuts across the 17 teaching hospitals associated with the Harvard system. And so there are faculty from all of these kinds of different institutions who work together under one common roof. It's both a real roof, you're under that roof, and we have another roof 2 blocks away, and it's also a more conceptual roof because in addition to the work that goes on here in this building, there are about 100 faculty around town who are part of, members of this Broad Institute. What it's done is it's propelled a continuation of this theme of the Human Genome Project, taking it on to trying to find out information about cancer, understand all the things that go on in cancers, and inherited human genetic variation and how it relates to disease, risks of diabetes and other things. To be able to use these same kinds of tools to see if we can develop small molecule leads that might even someday point to drugs for particular diseases, and many, many, many types of things like that. And it's just extraordinary to watch a new young generation of scientists come together in this world, and particularly in this city, who view themselves as part of this extended community, whose allegiance is not to, you know, their particular department or their particular institution. It's to getting some problem done, like really making progress in cancer. They're willing to share in all sorts of ways to do that. It's an honor for me to be a part of that whole community and see the energy and how much is happening and how far we've come, even in the 5 or 6 years since the official completion of the Human Genome Project. When we talk about communities, we do talk about a community of scientists or a scientific community, but we mean the sense of community much more broadly than that. The Broad Institute has a commitment to the community at large. When we produce our data, it goes out freely on the web to anybody. And when we talk about the scientific community, we don't necessarily mean just those people with PhDs or scientific training work at the bench. we really mean the whole community of people who are going to be affected by and therefore should be involved in the kind of science that gets done here. And so an important part of the Broad Institute has been the sense of obligation to our own community in which we live, here in Cambridge and the greater Boston area. There are programs here at the Broad for high school students. There are dozens and dozens and dozens, I don't even know the number of high school group, Megan will know, that come and visit the Broad from their high school classes. There are groups of high school students who take part in Saturday classes here to projects, about 15 students each term. There are summer interns we have. We have, as you see in the lobby, the rudiments of, and we're working on it, a little bit more of a kind of open space. It's not exactly a museum, it'll be called the DNAtrium. And so we're working on filling out a DNAtrium there where people can do it. And we come and sort of see what science is about. And one of the traditions that started now 3 summers ago was putting on a Midsummer night's science lecture here at the Broad over the course of the summer for several weeks. And we've asked various members of the community to come and speak and share their work and their excitement about what they're doing with whoever comes in. And you're whoever came in, and it's great because it's open to anybody who feels like coming. There's no assumptions about what you necessarily know, and it's really meant to be to share. And so I could not be more delighted by our first speaker in this series. Dr. Ann Carpenter is an extraordinary scientist who runs something called the imaging platform here at the Broad Institute. She is an expert in cell biology, but she's also an expert in technology who knows how to use high resolution and high throughput images, photographs, taken from all sorts of cellular situations to extract information about cellular processes and about disease and about molecules. And I gotta say that 10 years ago, nobody even thought of doing anything like this, looking at billions and billions of bits of data to try from images to extract information in this way. And Ann is really pioneering an important new direction and something that I think, well, the fact that she collaborates with 60 or 70 people around our community just indicates, how empowering it has been. And she's also a wonderful speaker and a really enthusiastic scientist. And so I was delighted when Ann agreed to give the first talk. And she will speak about robots versus disease. The robots work for her. Ann, thank you very much.
Sure thing. All right. So I'm hoping to convey some of the enthusiasm that Eric introduced you to about the work that I do in my group. And in fact, actually, I think we really want to bring out that the kinds of projects that I'm going to tell you about that we do in my specific group are really just examples of the kinds of things that are going on in science worldwide these days. It seems that science is becoming much more large scale. The projects that we undertake require a lot more resources and expertise in different areas that could just never be done in an individual laboratory. So no one has the expertise to do these kinds of projects that I'm going to talk about today, and you'll see some of the examples of what our own group has been doing here in the Broad Institute. So as Eric described, we are in a very special environment in the Boston area. It's I think, the center of the world for life sciences research. And here in particular, we have the ability to work together with faculty from MIT and Harvard and several other institutions in the area. And especially, these faculty are professors at some of the best research institutions in the world, but many of them are also physicians in some of the hospitals, the Dana-Farber and Mass General, Brigham and Women's Children's Hospitals. So these faculty come here to the Broad usually once a week to meet in different scientific areas, and this almost looks like the listing of a pharmaceutical company. They have different programs that meet together here. The faculty come together, they spin off ideas of what kinds of projects would be good to do that are probably not suitable to be done in any one individual lab, but instead require some advanced technologies. And so the technologies that are housed here at the Broad are listed here. I'm going to tell you about my own area of expertise, imaging. sequencing, I work very closely with chemical biology and RNAi, but I wanted to start my story today actually going back to sequencing. So Eric alluded to the fact that that was one of the first projects where scientists around the world really got together on a project of enormous scale that was really important for humanity and for providing a foundation for a lot of the science that I'm going to talk about today. So we'll begin there with how have robots helped out in the past. So robots have helped out a lot with sequencing, and first I'll describe what sequencing is. Of course, so this is a little snippet of genome sequence that I made up, and we read off the letters that make up the genome by looking at the DNA double helix. So this is a very iconic structure. I'm sure you've all seen it, and the little ladders that make up the DNA helix consist of a sequence of letters. And so when we say we sequence a genome, it means that we determine the letters that make up the entire sequence. That DNA is packaged up into chromosomes, and you'll probably recognize what those look like as well. The chromosomes are packed up inside the nucleus, and the nucleus is contained within a cell. So sequencing started out very slow. So Eric said it started a lot of the idea of, hey, is it even possible to sequence the entire human genome? The NIH began some sequencing trials in 1990 using a technology that was pretty expensive and pretty slow. Now the reason I've chosen how many days does it take to sequence 3 gigabase pairs is because the human genome is roughly 3 billion base pairs. And so given the technology in 1990, this was going to be an enormously expensive project and would take a very long time. But I think as we've seen advances in technology helping us in the computer sciences as well, they've been very helpful in biology. And so, okay, so then when I started my PhD in 1997, I did some of the sequencing myself, and can I just tell you, it's an incredible pain. So first of all, it's radioactive. Basically you pour this layer of jello that's not more than 1/10 of a millimeter thick. You try to have the thing not fall apart as you're reading off these radioactive bands. And so at that rate, nothing ever would have gotten done. But thankfully, around 2002, this instrument came out on the market that took together a lot of advancements that had been going on in the field, bringing the cost down substantially and improving the speed substantially. And this was the workhorse that really did most of the human genome sequencing. As Eric said, a lot of it down the street at 320 Charles, where the other Broad site is located. So that really improved things a lot. And what I'm really excited about, not going to talk about it all today, is next generation sequencers, which are coming out this year. Many of them are being tested here in their beta versions. But the cost and the speed improvement is so substantial that it will not be unusual to sequence a human genome in a week, which is just phenomenal. So this is showing you what that looks like. This is the sequencing floor at the Broad at 320 Charles. And it shows a lot of the instrumentation and the robots, and yeah, a few people as well who are involved in that process. Actually, I believe there's probably more people involved on the computational side of what do you do with the reams of data that pour off of these instruments than actually operating the instruments themselves. So that's how robots have helped get us the human genome sequence. And there we go. How has that actually helped us? So this, again, is not my area of expertise, but it doesn't take an expert to tell you that you may have seen press releases from time to time saying that, hey, we've discovered a gene that's related to a particular disease. It comes out in the papers, everyone's really excited, and so that was happening for a while, a number of different papers. The full human genome sequence came out here, and that provided the foundation such that in more recent times, the number of genes that have been associated with diseases is just phenomenally increasing. So this, I think, is a great triumph of this method of doing science in a big scale way across many laboratories and sharing the data freely amongst groups around the world. Okay, so we've used the robots to sequence the human genome, and we've also been using the robots to identify particular genes associated with particular diseases. But now what we need to do is figure out what are the functions of all those human genes, and most importantly, what are the functions, how do those genes go wrong when we end up with disease? So what are the actual mechanisms that relate to each particular disease that human beings are prone to? And that's a pretty big project, and one that my group is very interested in. And so does anybody know the central dogma of biology? DNA makes RNA makes protein, yay. So DNA makes RNA makes protein. So the DNA, this is the genome sequence that I've been talking about. It makes a material called RNA within the cells that makes proteins, and the proteins are really typically what gives you the end result. So if you have a gene mutation that leads to a particular disease, it means the RNA is a little messed up, the protein's a little messed up, and it means that ultimately your cells or your organs are messed up in some particular way. So what we would like to do, and the foundation of most of the experiments I'll tell you about today that my group's been working on, involve two different technologies. One is called RNA interference, and that's a technology where we can take, if we're interested in a particular gene, we make a particular RNA interference reagent that specifically knocks out that RNA. And what that means is that if we don't have this RNA, then suddenly we don't have the protein, and we can see what happens to the cell. So it's something like saying, well, if we mess up this gene, what happens to the cells? So that's one technology. The other one is using chemical compounds. So chemicals, sometimes chemicals are very specific to a particular protein, and sometimes they inhibit the function of that protein. So we can also use a library of many different chemicals to identify which chemicals interfere with which proteins, and then look and see what happens to cells. All right, so let's so if we're going to do those experiments, what that means is that we have to take one RNAi reagent or one chemical and put it into a dish with cells and see what happens to the cells. And that is a pretty straightforward thing to do. When I started my PhD, I had a pipette just like everybody else, and I cannot tell you how excited I was about halfway through when we acquired a super amazing multi-channel pipetter. That was a real triumph for me. But now it's actually not at all unusual to have these robots. There's a number of them here in this building, and you can actually see outside in the main lobby several of these kinds of instruments that are designed for pipetting, usually at least 96, if not 384 samples at a time, sometimes 1536. So quite a lot of samples can be processed with these little robots. This is what one looks like in action, and you can see, again, some out in the front lobby that are going. Okay, so these kinds of things make it very straightforward to prepare a lot of samples very quickly. So each little pipette has a different chemical, or each little pipette has a different RNAi reagent to test a different gene, and then all the samples are tested like that. Okay, so now we have all these samples. We still have to look at them, usually by microscopy, because the cells, of course, are very small. So here my timeline's a little more drawn out. We're starting in 1660, going to 1996 pretty quickly. Suddenly, we have an automated high-throughput microscope. I actually automated my own microscope during my PhD project, so couldn't be that hard. And so now it's actually becoming quite common to get these instruments in laboratories. So now we can start thinking, okay, now we can prepare hundreds of thousands of samples using these robots. We can image hundreds of thousands of samples using these robotic microscopes. What kinds of experiments can we actually do with these kinds of technologies? Well, for one thing, we can test, I just have a couple examples here. We could test 1 million chemicals to see which ones impede a particular type of cancer. These are the kinds of experiments that are especially common in pharmaceutical companies, but are becoming more common in the academic world as well. We can test chemicals to see which ones prevent neural degeneration. We can test every gene in the genome to see which ones are required for proper metabolism or any other biological function of interest. In essence, we can now essentially conduct any microscopy experiment across the entire genome or across 1 million chemicals to identify which samples are interesting. Now, the only problem with this is that we would have to do a lot of outsourcing to have somebody look at these 100,000 images. So it's great to produce 100,000 images a day, but how on earth do you actually look at them? And that's where my group comes in. The imaging analysis is our area of expertise. So a typical experiment that we work with, and I'll show you a few examples today, typical experiment involves a biology research group somewhere in the Boston area or around the world, somebody who comes to us for help. They say, I have this really interesting problem. I have a particular type of leukemia that I'm trying to address, and I want to find a chemical that is able to inhibit the leukemic cells from affecting the human being, so that it might make a good chemical to put into clinical trials. So they figure out what kinds of cells they need to put in the plates. They'll treat each sample with a different either RNAi gene perturbant or a chemical perturbant. And then they will set these on the robotic microscopes and end up with these images. And where my group comes in is we write software to analyze the images of the cells. We basically identify every compartment of every cell, and we make a lot of measurements of those cells. And then we work on data mining techniques to really extract the quantitative information from those images. Long story short, our goal is to take, for each image, we produce some measurement of what it is the biologist is looking for, so that we can rank order the samples that have been tested and identify those samples that are most going to be interesting to follow up on. So most of the time, the end result of a big experiment that we do, we test 1 million chemical compounds. Our goal is to come up with the top 5 that look like they might be effective against a particular type of leukemia, or might be useful in treating Alzheimer's, for example. So that's the basic outline of the kinds of experiments we do. And I'm going to start with a case study on tuberculosis. It's a worldwide health problem, not just the initial infection, but because it's very common for the disease to be latent and contagious and very resistant to antibiotics. As I'm sure everyone's aware, antibiotics in general, we need new classes of antibiotics because the old ones, the bacteria, are sneaky enough to get around. So we began working on this project with Deb Hung, who is a faculty member here at the Broad, and she has a postdoc in her group named Sarah Stanley. And in order to they came up with an idea for how to address this world health problem using the kinds of technology that is in my area of expertise. So in order to understand their goal, you have to understand traditionally how are antibiotics discovered. So normally what's been done in pharmaceutical companies over the past decades has been to put bacteria, and I've represented them here as green, in individual wells of these multi-well plates. So each one of these is a little test tube with bacteria growing in it. They're green. And they'll add 1 million test chemicals. Each chemical goes into a different well. And the goal is to find the 1 well out of 1 million, or the 5 wells out of 1 million, where suddenly the bacteria are dead. So that's great, except that sometimes the chemical also kills human cells, which is not great. This is a great approach for finding an antibacterial soap, for example, because it's killing bacteria in a dish, in a surface, but it's not necessarily terribly effective in human beings. And so what you'd really like to do is come up with a more physiological system, especially for the latent forms of tuberculosis that are sort of lingering in human beings. You want to figure out a way to get rid of those. So the effective but not ideal way to get around this is, why don't we take a more physiological system? Why don't we take a bunch of little girls and feed them 1 million different chemicals? And over time, our goal is to come up with some method to measure how much bacteria is in each girl and find the one that doesn't have very many bacteria. But if that's too complicated to come up with an assay to measure bacterial levels, we could just wait a while. So as scientists, we're kind of sensitive to the whole evil scientist thing. So actually, they came up with a different method, and this is what it is. It's not evil, and it's still pretty effective. So instead of taking human beings, we can at least take human cells. Those are shown here as blue. We can grow them in dishes. We can infect them with tuberculosis, which are actually these green blobs that are infecting the cells. And then we can look for chemicals that cause this effect. The human cells are still growing happily. They're not all shriveled up and unhappy looking, but the bacteria are gone. So that can work by 2 mechanisms. It either killed the bacteria, or it prevented them from infecting the cells. Either one is a really good approach, and it's likely to be a different mechanism of action than former antibiotics that were discovered by other mechanisms. So here are the folks that are working together. I'm going to definitely emphasize the collaboration throughout the talk because that's just how science is done today. So this is Sarah Stanley. She's a postdoc that I work with, and she's in Deb Hung's group. So this is what their experiment looks like. You put the bacteria and the human cells in individual wells. You treat with all the chemicals, you run it through the robot microscope, and then you end up with these 100,000s of images. So anybody spot the one that is the hit in the screen? So this one right here has no bacteria. Unfortunately, without some kind of automation, you have to start cloning postdocs in order to identify that. So that's where my group comes in, and I'll show you how this works. So what we want to do is we want to feed each image to a computer. We want the computer to tell us to find the individual human cells. We want it to find the clumps of bacteria, and we want it to count up how many bacteria are present per cell. And so this is where we get into the technical details of how the image processing actually works. And I think it's actually relatively understandable. A lot of you are probably into digital imaging and have some sense of how the images that come off of your digital camera look. This is a black and white image, but it's the theory is pretty much the same, that you have individual pixels, I'm sure you've heard that word, where, if, for example, I've zoomed in on a region of the image here, and you can see the background is mostly zeros, and the cell here is mostly a very high number, 255, and then you have all these intermediate values in between. And so that images really are these just collections of numbers. And if it's a collection of numbers, then we can do math on it. And I'm going to describe some of the simpler algorithms that we use, but I think you'll get a picture of how this works. And if you bear with it, I think you'll be quite surprised at how straightforward this is. So first of all, that was a really nice looking image, but this is what we tend to get. So there's sort of dark background here, and there's light background up here. It's kind of hard to count individual cells when you've got these variations. So the first thing we have to do is clean up the image. And the way we do that is we run a mathematical filter across the image. And it might sound a little intimidating, but the only thing we're doing is we're running this little box across the image, and within each square, we're asking the computer to tell us what is the lowest value that you find within that square. So up in this corner of the image, the lowest value might be 10 or 15. Whereas here, where the background is very bright, the lowest value might be something like 50, right? So within each of those little squares, you just ask the computer what's the lowest number within this square. That's pretty straightforward. The computer can do it very quickly, and it produces an image like this. So the result of applying that math to that image is this. And that gives you a kind of that overall background. And what you can do next is just subtract these two images from each other. And it results in this. And so when I say subtract, I actually mean subtract. You take, some of these the bright things are 255, and you subtract this background value at that same position, and you end up with a nice flat background. Does that make sense? It's actually very simple math, and the computer can do it really rapidly, and it gives us a good starting point for cleaning up the image. And so now, with this very nice black background, you can see the scale bar here. The background's usually 0 to 5 or so. The objects themselves are something like 50. So now you can say, computer, go and find all the pixels that are above 50, because those are probably cells, and find all the pixels that are below 50, and those are probably backgrounds. So that works pretty well. And I'll show that schematically here. So here's an original image, and you can look at it in kind of a three-dimensional way. It's a little easier to see what's going on. So if we say, show us all the pixels that are above 50, it's sort of like making a horizontal slice across this image, right? We're just sort of chopping off the little mountains there and saying anything above that is a cell and anything below that is the background. So that's basically how it works. The problem is that you end up with something like this. So you chop at a certain plane, and you certainly get mostly cells are circled, and the background is mostly the background, but the boundaries are not that great, and you get things that are stuck together. So you think, okay, well, these two guys, I don't want them to be stuck together, so I must have sliced too low. I should slice higher where the two peaks are actually separate, and that way they'll be separated into individual cells. The problem is you do that, if you slice high enough to get those guys to be separate, then suddenly you miss this guy, this little guy, and this guy all together, and you don't find them at all. And as well, the outlines don't become accurate at all. Okay, so how do we get around this? We used an algorithm actually from somebody's PhD project over in Europe. We got her thesis online, and it seemed like a really good approach, so we used it. This is basically what it does. It says, it again runs one of those filters across the image and says, within each square, find me the local maximum in the image. And so it's pretty easy to run that filter across the image, and it identifies basically the peaks of every mountain. So it only calls something a peak if everything around it is lower than it. And that gives you the center points for all of these individual cells. And then you run something called a watershed algorithm, which is named after the fact that you pretend you're shooting water out the tops of each of these little volcanoes, and it's running down, and basically where the water hits another cell, that's where you stop. So these two guys, for example, the water shoots out the top of those two guys, and where it creates a really nice dividing line between them. And as you can see, the results are much better. So you're not only, you're getting all of the individual cells, but you're separating them properly. So even if they're clumped together, you're able to separate them. All right, so that's how it's done. If you totally blanked out for that section, you can come back and join us, because this is what it looks like. So running those algorithms on this type of image yields this. And so you can see even when the cells are really clumped together, we're able to separate them into 2 individual cells. And we do the same thing for the clumps of bacteria. We identify those and kind of assign each clump of bacteria to a particular cell, and then we count up the bacteria per cell. And the person in my group, Martha, who worked on this, works closely with Sarah to work out these conditions, and they are getting to the point where they're going to be starting up one of these screens. So we've got it working, and we'll be testing chemicals in the next few months to find which chemicals are able to get rid of the bacteria but leave the human cells intact. All right, so the software, as I mentioned, is open source, which means that it's freely available to scientists all over the world, and you can go there yourself and download it and take one of these examples, and this is showing you what it actually looks like. So this project, I should say, began as a collaboration with Ray Jones. He was a PhD student at the Computer Sciences Artificial Intelligence Lab across the street at MIT, the crazy data center building that leaks. You may have seen it. So he helped me get started on this project, and together we wrote this software over the past 4 years. And this is what it looks like when it's running. You have this pipeline of modules, and it runs on every image in the experiment. So if we have 100,000 chemicals, it'll cycle through 100,000 images, identify all the individual cells, and then measure all their properties. So people use it all over the world. We especially have, for some reason, a pretty fanatic fan club in the Netherlands and in Switzerland. And I'm going to speak to the group in Switzerland next week. People all over the world are using us, and what we really like about it is that it can be used not just for the robotic intensive experiments that I've described here, which require, of course, a lot of resources to pay for the instrumentation and the engineering, but they can also be used for very small scale experiments. And I'll use as examples of the small scale experiments that we can do. It's very common in laboratories that are studying yeast to test a bunch of different samples, chemicals or genetic mutations or something, and try to identify which ones are not growing as well as the others. So this used to be done just by eye. You would just say, well, this one and this one and this one and this one and this one are not growing as well. But with this kind of, using the same algorithms that I showed you for cells, we can quantitatively get numbers out of each of these samples. So how well is it growing? Not just yes or no, is the spot there or not, but exactly the numbers that are involved. We can also do another experiment, which is very close to my heart, because my very first, I spent a summer in Iowa counting yeast by eye. So this is something that is the kind of experiment that's done all over the world. It's very inexpensive. It's done in a lot of teaching laboratories, especially. In this case, we used it for a project that was looking at pathways leading to drug-resistant yeast, which is a very big human health problem. But basically, these are agar plates with little yeast growing on them. And now all you have to do is slap them down on a flatbed scanner, take a digital image of them, and then use the software to identify all the colonies, and then you can classify them according to how pink are they, how white are they, how big are they. All that kind of information can be done. I know it doesn't look like much, but this is something like 1,000 colonies and is really, really boring to count. Even if it's air conditioned in Iowa in the summer, it's still really awful. Okay, so back to the main thread. What I wanted, the really cutting edge thing that we've been working on lately is not yet published, and it's pretty exciting because it's opened up the door to do a lot of experiments that we just couldn't have dreamed of even a year ago. And so as background, I'll describe why we decided we needed this technology. It's because we were looking at these experiments that use RNA interference. So we were knocking out each gene, and we happened to notice some really funny shapes of cells that resulted, different genes that we'd knock out. We just were browsing some of the images, and we saw some really funny looking cells. So this one I thought was particularly interesting because it was a particular gene that we knocked out, and the cells looked all crescent shaped. And looking into the literature, it looked like this particular shape of cells was often seen in a particular type of colon cancer. So I really wanted to find all of the genes in the genome that resulted in cells having this shape. And as well, I wanted to find all the genes in the genome that caused this shape and that shape and this shape, and a bunch of others that are not shown here. So in order to accomplish this, the problem is that previously I've just showed you how we've counted things and how we've measured intensity, so, right? So you just sort of measure how bright is something, how many of them are there. For this, we needed to measure how does the thing really look, combinations of features that tell you how the cells are actually looking and measure them so that you don't have to look at every image by eye. So again, Ray Jones came through here with a really great solution, and it involves supervised machine learning. And what we do is, again, we have these thousands of samples. We use Cell Profiler to identify every cell in the image and to make all kinds of measurements of those cells. So I'm going to represent schematically. So here's a little cell that we've measured in all sorts of ways. And here are the different features that we've measured. And we call it a cytoprofile. In reality, we have like 500 or so different measurements that we make of every cell. And it's just shown here schematically just to better explain how it works. So what happens is the system gives a few hundred cells to the biologist to score. And so the biologist sits and looks at these images and decides which ones look like what she's interested in and which ones are not interesting. So in this case, it looks like she is scoring for cells that have some kind of little projection, but not pointy ones. They have to be blobby projections. So she's just saying these look like interesting and these are not interesting. It doesn't matter. So each biologist picks whatever it is that they care about. And so interestingly, the biologist is looking at the pictures. She doesn't see the numbers or care about the numbers. The computer doesn't look at the images at all. The computer's looking at the numbers. The computer's looking at all the measurements that have been made of these cells, and it tries to figure out what do these cells have in common that's different from what these cells have in common. And it figures out that some of the measured features don't matter at all. So for example, it doesn't matter how big the nucleus is or how big the area of the cell is. It just matters that these have a certain blobby shape and these don't, and there might be other features. So the computer starts to get an idea of what the biologist is looking for, even with just a handful of cells. And then we go through this process iteratively so that it says, okay, I think I know what you're looking for. Here's some more cells that I think are positive. What do you think? And the biologist says, yeah, most of those look like what I'm looking for, but some of them don't. And so she sorts more, and then the computer figures out better what she's looking for. And this process goes around and around until it's quite accurate at identifying the individual, the appearance of the cells that she's looking for. So once that's done, the computer has a really strong idea of what the biologist is looking for, and it can then score all the cells in the experiment. So if we have 100,000 images, that means we usually have millions of cells to be scored. And so as each cell passes by the rule, it compares how does that cell match up to what the biologist is looking for. It gives it a score. And then we just add up for every sample how many positive cells are there and how many negative cells are there. So that's how the machine learning works. So it's released in another software package that we just put out a few months ago. And this is what it actually looks like. And I have to tell you, it's the most fun thing in the world to play with. So you start it up. You say, show me some random cells. And in this case, I'm going to sort for cells that have this green bar-shaped nucleus. And so I'm putting the green bar-shaped ones over here in positives and anything else over in the negatives. So this one was too round and blobby. I only want the bar-shaped ones, so I'm sticking them over there. So I have to sort a lot of them. Actually, you only have to sort like usually 20 or 30 before it gets a good idea of what you're looking for. Then you train the machine learning, which means you push this button, and then a bunch of gibberish comes out. You have no idea what it means. And then you say, give me more positive cells. And this is what it does. It says, I think I have a good sense of what you're looking for. These, I think, are positives. And if you disagree, you can keep sorting until it's really perfect. And then you score all the images from the entire screen. And it says, OK, you had 100,000 samples. Here at the top of the list is the one that has most of these cells that you're looking for. And so you can open up that image and see how it looks and confirm it by eye. So as biologists, we always want to go back to what the sample really looks like to our own eye, even though we trust the computer to sift through the hundreds of thousands for us. So this is an example of a good hit in the screen. These cells usually only occur maybe one in an entire image, and that one had 15 or 20. So that was a really good hit from the screen. Then we go and we figure out what gene is it that caused that particular unusual shape of the cells. So that's how it works. It's really fun to play with. The biologists love it. And it allowed us to score hundreds of thousands of samples for each of these different unusual phenotypes. It worked really well for all of them. And what I'll close with is showing you, I have 8 different examples here of different groups that we've been working together with using this technology in a lot of different biological areas. So in a project related to glioblastoma, which is a particularly aggressive form of brain cancer. We have a project ongoing with leukemia and different cell division, which is related to cancer progression, projects specifically in breast cancer. You can see how many people are involved in these projects, and these are people across the Boston area. Some of them are physicians, some of them are biologists, some of them are our group who are computer scientists, software engineers. A lot of collaboration going on to get these kinds of projects done. Again, more projects related to cancer, leukemias, and radiation oncology treatment. So what's up next for us? We're excited to be expanding beyond just individual cell experiments to things like really unusual cells like neurons that have these long pointy projections. We're doing experiments in whole organisms, so we use the little worm C. elegans for experiments. C. elegans is great because, as you can imagine, it's not that easy to study things like behavior in individual cells. You can't study, so in these guys you can study how do the worms respond to starvation? How do they move through the dish? How does their muscle function work? And those kinds of things, which you can't do in individual cells. Same story for zebrafish. There's another good model system where you can use the whole organism to study things. And as well, we do some work with time-lapse images, because a movie is really just a pile of images. And so we can identify objects and measure them over time using the same kinds of technology. So the people in my group who have been most helpful in all of these projects are shown here. It's a really great group of people that we've recruited just in the past year since I established my group here. And with that, I will conclude, and thank you very much for your attention and for coming today. So I'm happy to answer questions if there are any.
This technology seems to be free and available all over the world. Most people seem to be using it for very helpful and productive and positive uses. Do you worry that there might be some that would use it for frightening or dangerous purposes?
That's a good question that you could ask really for any type of technology, whether it can be put to bad purposes. I think in this case, probably I don't think it's any different than any other technology, that it could be put to bad purposes. For example, engineering more infectious bacteria rather than trying to cure them. Yep.
When the biologist picks out the sort of target type of cell and stuff, would you have disagreement among biologists?
Oh, that's the only thing.
The rest of it I thought would be, oh, it's so user-friendly, I just love to play with that. But would they all agree, you know, on like the sickle-shaped one or something, that's the thing you look at?
Yeah,
throw out this other stuff.
That's a great question. So the question was, do individual biologists agree on what counts as a positive and what counts as a negative? And the answer is absolutely not. And so it's very important whenever we do the test to try to determine, is the software doing as well as human beings? We always have to have at least 2 human beings. And so over the past couple of months, I've had a good number of my very gracious friends scoring cells in their spare time to try to figure out what is the agreement between individual human beings, because we can't expect the computer to, there is no absolute truth in terms of whether a cell is crescent-y shaped or not. There's a smooth gradient. Some, you know, everybody decides what they think is crescent-y and what's not crescent-y. So that's a very important thing to think about when we test the software.
How many cells do they have to score in order to get good supervision?
So it depends how simple the phenotype is. So for the metaphase one, I showed hundreds of cells on each side, positives and negatives, but since then I've done the experiment with, it usually only takes about 40 or so on each side, and it gets that phenotype really well. For some of them that are more subtle and more complex, or that are just more rare in general, it might take, I think the largest one we've had has been about 1,000 cells total across the 2 sides. In any event, it sometimes takes as little as 45 minutes and sometimes as long as an entire day. But given that, as you can only imagine how long it takes to set up one of these experiments, it's usually a 6 to 8 month project. So spending a day sorting cells is usually not too bad.
Have you used the cell profiler software to target cells in tissues, complex tissues, and 3D scaffolds?
So the question is whether we have used these kinds of methods on tissues and three-dimensional structures, and we have not yet. But there are a lot of as I described, we got some of our algorithms from people's PhD theses and from literature, and there actually are a lot of algorithms that work very well for 3-dimensional images. that we just haven't incorporated into the software yet. So we actually just today hired a software engineer. So, so I think some of those things will be coming down the line very soon. Yeah. Here.
Image analysis is multidisciplinary. When I saw that plate of yeast, it reminded me of some astronomical images as a scan. So I know that there's a lot of image analysis, pattern analysis in that. Have you looked at, has someone looked outside of biology for other uses of, or where other uses are?
That's a great
image
question.
analysis?
So the question is whether the same kinds of technologies can be used for astronomy or for other fields, because especially the yeast, for example, look a lot like stars or vice versa, right? So the funny thing is, so we are definitely, right now, biologists are on the receiving end of this kind of image processing technology, because it's only been applied over the past 10 years or so at all in our field. So we're not producing the cutting edge things and giving them to other fields. In fact, about a year ago, I went down to Washington, D.C. to a government agency that was interested in, it had a lot of astronomers and physicists in the audience, and I described to them what we were doing, and they were just so sad for us, because because in astronomy you don't have a lot of images to work on, right? They collect the images and they're all given to the scientists, and then they crunch away on them for years. And so they've spent decades and decades perfecting methods to just get the most out of every image that they have available. So the Hubble Space Telescope goes off into the wild blue yonder, and that's all you've got to work with. So so what's really interesting is that we can leapfrog quite a bit based on what the other fields have done so far. And so far, we're definitely still, we're probably still catching up to where astronomy and some of the more physical sciences have been for a while.
It's not obvious, of course, how human scientists score your profile. So have you ever talked with a cognitive neuroscientist about what your approach might teach them, or what they might teach you?
That's an interesting question. No, I guess the simple answer is no. We haven't really worked together with people who are interested in visual perception that human beings have. But it's a really interesting question, and certainly MIT is a good place to find such folks. I think there was a question up there.
What you described is neural net as far as science is concerned. Have you ever worked, once you set up the profile and the computer thinks it knows what you want, have you ever worked backwards to see what features the computer is zeroing in on?
Okay, yeah, so the question is, once we've trained the computer, do we ask the computer, what exactly is it that you're looking at? And the answer is yes. So when I was joking that it spits out a bunch of gibberish and no one knows what it means. Well, okay, actually we do know what it means. So if you play around with this stuff long enough, you can read off what the features are that it's taking into account. And sometimes it's actually led to some surprising results. So the crescent nuclei is a great example where what we were seeing was the crescent shape of the nucleus. So we trained the computer, and then we looked at the features that it was using. And of course, some of the features had to do with the shape of the nucleus. But we were really surprised that some of the very top features had to do more with how the outer boundaries of the cells looked. And we hadn't noticed that the outer boundaries of the cells were unusual at all. But once we saw that that was a feature the computer was looking at, we went back and looked at the images and noticed that it was really true, that there was an association between the funny shaped nucleus and as well the boundaries of the cell. And so the computer in that case was teaching us. Maybe that's a good place to stop. Thanks everybody for coming. There's reception out here.